session through April 13th, where we're going to get Rena Das Eiden from Penn State University come in and speak in conjunction with our co-sponsors here at C-StARR. And here's the tutorial.
Good afternoon, everyone. On behalf of my colleagues in the Center for the Study of Adolescent Risk and Resilience and the Center for the Study of Addiction and Behavior, I'm pleased to introduce today's speaker, Dr. Elizabeth Lippard. Dr. Lippard is Assistant Professor, tenure track in the Department of Psychiatry and Behavioral Sciences in the Dell Medical School at the University of Texas at Austin. She earned her bachelor's degree at NC State and her PhD at UNC Chapel Hill in neurobiology. After completing her postdoctoral appointments at Yale, she joined the faculty at Texas, where she is now. Dr. Lippard directs the Mood Addiction Interaction Neuroscience Lab, in which she provides research supervision for trainees ranging from PhD and med students to high school students. Her team's work has been funded by multiple grants, most recently from the National Institute on Alcoholism and Alcohol Abuse, from which she is currently funded by a Mentored Research Scientist Development Award. Her productive research program focuses on the neurophysiological mechanisms contributing to the onset of mood and substance use disorders and interactions between these conditions. In addition, she's involved in efforts to develop innovative educational curriculums with a focus on neuroscience and research at the medical and graduate school level. Please join me in welcoming Dr. Elizabeth Lippard.
Okay, can everyone hear me okay? So I've been told to project. Katie, I'm going to be looking at you. You give me the bring it up or seriously bring it down. It's too much, and I'll try to accommodate. I am beyond thrilled to be here. I was thrilled to get the invitation. Thank you guys. As you heard, I bleed red. I've been a light shade of blue. Hopefully we can get past our color scale differences. But no, it's been an absolute phenomenal trip so far. I'm looking forward to the rest of the trip. And I'm excited to share with you guys some of what I've been doing over the last roughly 9 years, including time whenever I was a postdoc, in the last 6.5 years as I've shifted over to the dark side and peeked behind the wizard's curtain to actually start a research program as an assistant professor, and specifically studying comorbid alcohol use disorder in mental illness with a focus on bipolar disorder and risk factors. So I'm going to give you today a very brief background, really focused on brain development, adolescence. We do have some adolescent researchers in the audience, but just to make sure we're thinking about why is it so important that we're thinking about development when thinking about risk factors for alcohol use disorder. And then I'm going to focus the talk on comorbid alcohol use disorder in the context of bipolar disorder and why this is so important just from an addiction perspective. I'm going to focus on disease heterogeneity, longitudinal work, and then the majority of the talk is going to focus on results from a placebo-controlled alcohol administration study in young adults with bipolar disorder, mixing in some future directions that I'm pretty proud of or excited for. So what do we know about brain development during adolescence? Again, very, very short and simplified, but we know that this is a dynamic time in terms of brain development. We know that there's synaptic pruning. You see decreases in gray matter volume and the frontal areas of the brain. We know that connections continue to be made, to be refined, to be strengthened. You see increases in myelination, especially during the frontal limbic systems of the brain. And we know that this healthy development is associated with more mature and adaptive behavioral responses. So adolescents, as these systems develop and come online, they become better able to regulate their emotions, better able to regulate behaviors. We also see that they're better able to control their impulses and risky decision-making. We also know that altered development during this time within these systems coincides with increased risk for mood and alcohol and substance use disorders. So what are the regions of the brain that's implicated in regulating emotions and impulses? I don't have to go into this in detail. This is very simplified, but we know key players are the amygdala, the striatum, and the frontal cortex, especially this ventral prefrontal cortex. And as I mentioned, during this adolescent young adult period, this system is coming online in which connections are continuing to be refined, to be strengthened. And so as the system comes online across adolescence, the frontal cortex begin to act as a brake, basically telling the limbic and the striatal system, wait, we don't need to be this active about this context. It's able to take all of these experiences and put it under a contextual content so that way we're able to regulate our striatum, our limbic responses to various environmental stimuli and various experiences. Well, bipolar disorder, which is what I study, is a disorder that's characterized by dramatic shifts in mood. Individuals will have periods of extreme mania or extreme highs, and then they'll have extreme lows or depression. Of interest to this talk, we know that bipolar disorder is associated with altered emotional regulation and impulse control. It's also associated with altered structure and function within these frontal limbic neural systems that you see here. And since this system is coming online during adolescence, there's a developmental hypothesis to bipolar disorder, in which bipolar disorder is thought to arise as a consequence of altered development within this system during adolescence and young adulthood. And indeed, adolescent onset of bipolar disorder is now increasingly recognized. So symptoms of mood disorders typically emerge right around puberty, so very early adolescence, with the first peak age of onset for bipolar disorder occurring between 15 and 19 years old. Now, keep in mind, bipolar onset continues, first episode mania continues through the 20s, through the 30s, right? But this is the first window when you really start to see manic-like behavior begin to emerge. And this highlights this period of adolescence as a critical time in risk, onset, and early disease course. We also know that the regional differences that's been implicated in bipolar disorder emerge in a developmental sequence. And by this, what I mean is that the studies looking at amygdala and striatum, these differences appear to emerge early and be stable, compared to other studies looking at the frontal cortex in which they appear to be much more progressive. And that if you look in older adolescents, the difference is a greater magnitude compared to if you're looking at younger adolescents. The same is also true for white matter. And so if you look at white matter, especially, let me see if I can get my laser pointer up, especially in regions of the frontal limbic system, like the uncinate fasciculus, what you see is that the magnitude of difference is much bigger if looking in later adolescence compared to early adolescence. And again, this supports this progression. It supports this developmental hypothesis of bipolar disorder. And whenever I was a postdoc, I was able to contribute to 2 longitudinal studies in bipolar disorder, looking at gray matter, white matter structure, also one study using diffusion tensor imaging to look at fractional anisotropy of white matter tracts. And again, seeing that, yes, adolescents, young adults with bipolar disorder show differences in neural trajectories during this critical developmental period, supporting this developmental hypothesis to bipolar disorder. So where does this take me to now? Well, during this time, what I was really interested in is that when you map this data and you look at within-person change, within-person trajectories, one size does not fit all. There is so much variability at the individual level. And for me, coming from an addiction side, I was very interested in understanding early disease heterogeneity, early neural trajectories, brain development trajectories that might relate to increased addiction risk, specifically in bipolar disorder. So let's talk about bipolar disorder and the life course. So this is a simplified life chart of one individual's manic and depressed mood episodes over the course of their lifetime. And this life chart is one individual whose bipolar disorder illness was left untreated. And what you can see, if you look at mania symptoms, they are your red bars going up, depressive symptoms or episodes are your blue bars going down. And what you can see is that if left untreated, symptoms get more severe, meaning they're moving away from this white line. But also their episodes become longer in duration. They start to occur more frequently. You start to see mixed episodes in which you see both depressed and manic symptoms present at the same time. However, there's many factors that can improve outcomes. One of them being treatment. Getting on the right treatment early in the course of the illness can have a profound impact. But we know that there's many other factors as well that can contribute to differences in disease progression. These factors include genetic variation, early life stress, specifically childhood maltreatment, which is something that I'm interested in, as well as alcohol and drug use. And again, I'm focusing on the alcohol and the drug use for this talk. So alcohol use disorder, it's important to remember that this occurs early during disease course in bipolar disorder. So again, if you look at this critical period during this adolescent young adult window in which risk profiles are happening, in which onset is happening, early disease course is happening, we know that on average first use occurs around the age of 15. And we know that symptoms of alcohol use disorder oftentimes emerge around the age of 19. And so again, this puts alcohol use initiation, alcohol misuse, the development of symptoms of alcohol use disorder at a key time in which we're seeing elevated risk for bipolar disorder. We're seeing onset and we're seeing early disease course that we need to be thinking about in terms of how do we shape treatments, interventions to improve outcomes. And it's not surprising that alcohol would have a detrimental impact on bipolar disorder. This is a list of the broad phylogenetic range of dysfunction that's observed in bipolar disorder. And just basically, we know alcohol affects every single one of these. So yes, it's going to have a negative impact. And indeed, epidemiological studies have supported that comorbidity is associated with more severe illness outcomes than either bipolar disorder or alcohol use disorder alone. This includes more severe mood episodes, greater cognitive deficits, more impulsive risk-taking, greater hospitalizations and health problems, and a high risk of suicide. And I should say this is a high risk of suicide compared to the already high risk of suicide that we know exists in individuals with alcohol use disorder alone, individuals with bipolar disorder alone. And so yes, while it's very important for us to think about how alcohol use disorder affects mood disorders and bipolar disorders, I would say it's equally important that we think about how mood disorders, bipolar disorder affects alcohol use disorder as well. And I say this because it is now well established that the majority of individuals with bipolar disorder will present with a comorbid alcohol use disorder at some point in their lifetime. Estimates are as high as 60% in individuals with bipolar disorder, which is 3 to 5 times higher than the rate that's observed in the typical population. And while the majority of studies in bipolar disorder have excluded individuals with bipolar disorder, again, we have to think about generalizability. Likewise, the majority of studies, especially those looking at risk factors, relapse, they exclude individuals with bipolar disorder. And so this is also equally important if we take a step back to thinking about why is it important that we think about how severe mental illness contributes to addiction. That's because mental illness oftentimes predates addiction. And these are factors that we need to be thinking about if we're going to be thinking about interventions that are much more specific based on an individual. These are just some data from NIDA, especially one that I'll highlight is this one right here that looked in adolescents that was in a substance use treatment program. And they found that 60% of these adolescents, so again, early individuals showing substance use disorder, 60% of them had a comorbid mental illness. So right, this is emerging early. We can't just sit there and think, oh, alcohol use is increasing risk for depression or increasing risk for these things over time. Comorbidity is emerging even early on in the course of illness. And so we have to be thinking about this other side of the coin. This is data from another study, again, looking at the prevalence of drug disorders. And what you can see here when looking at mania, you can see in this study, the higher prevalence of drug use disorders is found in individuals that showed a history of mania. So again, I'm showing this just to generalize and just to expand back and thinking, why do we care about how mood disorders might increase risk for an alcohol use disorder? And so why do they co-occur so frequently? Let me go back one. So we know that there's several hypotheses about this. One is causal effects. By this, I mean that substance abuse may increase vulnerability to mood disorders. And so changes in the brain ultimately may contribute to this comorbidity for subsequent mood disorders coming online. Also, self-medication hypotheses in which substance abuse may begin as a means to alleviate symptoms of mood disorders. There has been studies that shown that acute nicotine has antidepressant properties, while more chronic nicotine exposure has more of a depressive phenotype, just as an example. but also common and correlated causes. And by this, I simply mean that the risk factors that give rise to mood disorders and substance use disorders may relate or overlap. Also, just a note, I see here I have substance abuse. I'm continuously going back, trying to make sure I'm changing my language, removing abuse to be in line with terminology that decreases stigma. So if you do see that, just keep that in mind so that we all start continuing to try to use appropriate terminology that won't foster stigma around substance use. So one of the things that I'm interested in is thinking about comorbid neurobiology or overlapping neurobiology. And so what are the brain regions that are implicated in both mood disorders and addiction? And so I'm showing you these two pictures of brain regions that's been implicated, and it should immediately strike everyone why I chose to look at these two regions. It doesn't even matter what the regions are that they're highlighting. What matters is that there is substantial overlap in neural systems implicated in both of these conditions. And so a basic hypothesis is that increased risk for comorbidity comes from some of these overlapping neural systems that's implicated in these conditions, which is something that I'm interested in. Again, other common or correlated causes includes behavior, personality traits, as well as genes. Interesting side note, we know that there are common genes. We've seen genetic overlap in GWAS studies looking at alcohol use disorder, other substance use disorders, as well as mood disorders. But also transgenic rodent models, especially those modeling facets of mania, has shown increased alcohol and other substance use behaviors. Also factors such as stress, which is something that we're interested in. And so, but going back to the neurobiology, is there reason to think that differences in the brain may predate alcohol use problems and contribute to problems over time? Yes, there is. Several groups now have done longitudinal studies and seen that differences in the OFC, orbital frontal cortex volume, at 12 years is associated with cannabis initiation at 16 years of age. There was a study seeing anterior cingulate cortical volume at 12 was associated with alcohol problems at 16. Also, functional connectivity between the orbital frontal cortex and the amygdala in youth predicted recent and lifetime alcohol use 2 years later. And so, this is all done in the healthy, individual, typically developing youth. Now, one of the things I wanted to do when I was a postdoc was think, hey, well, can we actually predict initiation and problems in bipolar disorder? Let's extend this. And for those of you that are thinking about, you enter a lab, you're a postdoc, what are you going to do? I entered a lab in which they had a very rich cross-sectional data set of youth with bipolar disorder. And I was able to, while the lab wasn't interested in alcohol use disorder, they were interested in longitudinal studies in bipolar disorder. And so we were able to get some money so we could actually do a study. I was able to identify a very homogeneous group of youth with bipolar disorder. On average, they were 16 at the time of their scan, but follow them, reach out, follow up with them 6 years later to see which went on to develop alcohol and cannabis use problems compared to those that did not. And what we found, similar to the other studies in healthy youth, was that lower gray matter volume in the orbital frontal cortex, the insula, also rostral and dorsolateral prefrontal cortex, distinguished those that went on to have future problems compared to those that did not. Now, this was basically the study that got me started, that helped me figure out where do I want to go as I'm transitioning to having my own lab. And during this time, now what I had confirmed is that, yes, we may be able to use the brain as a marker of risk. We did some work looking at longitudinal progression in the brain and those that did have comorbidities, suggesting that comorbid alcohol and cannabis use disorder and bipolar disorder is associated with progressive changes in the brain, especially within these frontal limbic systems. And I also did a lot of suicide-related work during this time and suicide prediction-related work, suggesting that some of these changes may relate to increased risk for suicide. I'm not going to talk about any of that. Instead, what I'm going to talk about is where I went from there, in which what I wanted to do when starting my own lab was really to think longitudinally, to go back to the drawing board and make a much more quantitative physiological approach to studying the development of alcohol misuse, incorporating measures of early life stress, genetics, familial risk. And so that's where I started. And so in 2016, I joined as a faculty at the University of Texas at Austin. In 2017, I got my first longitudinal preliminary study up and running. We recruited individuals with bipolar disorder individuals that were at risk. And by this, I simply mean that they had a first-degree family member with bipolar disorder and also typically developing young adults. We did detailed clinical behavioral phenotyping and collected a battery of MRI data. And we were relatively successful to be brand new in a department starting to do bipolar disorder research. We did really great with enrolling individuals into this preliminary study. Again, the goal was longitudinal data. And so we were really doing great with our 1-year follow-up and our 2-year follow-up. And then something happened in March 2020. We won't go into it, but that was the end of the study at that point. But this study provided a wealth of preliminary data that's now been starting to be published, but that's also contributed to all my subsequent grants since then. And so one of the things, the first thing that we wanted to ask is, what relates to greater alcohol use in adolescents with bipolar disorder, right? And it's as simple as this. We saw that these regions were related to future risk. Well, if they are related to future risk, shouldn't they also be related to greater alcohol use in adolescence? Because alcohol use is going to predate the development of alcohol use disorder. And so we wanted to look at alcohol use patterns. And what was interesting, so this was from my graduate student, Valeria Tretiak, work that she did. And when looking at alcohol use patterns in these youth, we saw absolutely no difference in alcohol use. And this is really important to think about because this is a very generalizable cohort. These are individuals that had very prevalent suicide attempt history. They had history of psychosis. They were very impulsive in terms of looking at impulsivity symptoms, right? And this really challenges the notion that I've heard many times, well, we know why youth with bipolar disorder have greater risk for addiction. They're more impulsive. They drink more. They use more drugs. That related zip to any of what we were seeing right now within this data set. Now, that's not to say that impulsivity is not part of the story, but again, it helps us reframe. We need to be thinking about what are the risk factors early on that's contributing to initiation, early escalation of alcohol misuse before the development of alcohol use disorder. Now, one of the things that we did see is that our youth with bipolar disorder, more of them compared to our healthy comparison youth, reported more symptoms of alcohol use disorder. And so if we take a step back and we think about the stages of addiction. So for this study, we're trying to look before the development of alcohol use disorder. So we're trying to look when individuals drink for more positive reinforcement effects of alcohol use disorder compared to later after the development of alcohol use disorder when you see this dark side of addiction and individuals are drinking for negative reinforcement. And so the simple hypothesis was, okay, well, if they're not drinking more, but they're showing more problems and symptoms of alcohol use disorder more, are they experiencing alcohol intoxication differently? Is the way alcohol affects the brain different in the context of bipolar disorder? And is there any support for this? Yes, there is. So Sarah Yip in 2012 published a paper looking in young men with a broad bipolar phenotype. These were not individuals that had a bipolar disorder diagnosis. They had just had previous hypomanic-like symptoms. But what she found is that these young men, shown here in the gray bar, reported feeling less high when drinking alcohol compared to a healthy comparison youth sample. Now, there's also, went one too far, there was also this study that came out of John Crystal's lab. So very few studies have looked at alcohol sensitivity, response to alcohol in the context of severe mental illness. This is one of the only studies that has done that, and this was out of John Crystal's group. And they looked in adults with schizophrenia. And what they found is that in schizophrenia, these individuals, adults, are reporting greater sensitivity to alcohol, reporting that they feel more high when drinking alcohol compared to healthy comparison adults. Now, while these two findings support two different models of subjective response to alcohol, one being the low response model in which people don't respond as much to alcohol, so they drink more to feel intoxicated. There's a lot of empirical support that, yes, this can increase risk for alcohol use disorder. This is Mark Shuckett and his group. There's also the differentiator model in which individuals can have greater sensitivity to the stimulating effects of alcohol, but lower sensitivity to the sedative effects of alcohol. Also a lot of empirical support for this as a risk factor for development of alcohol use problems as well. Andrea King and her group are some of the leaders in this model showing this data. But what these two studies converge to support is that youth with bipolar disorder may show differences in sensitivity to alcohol, and it may relate to risk. And so this was one of the things we wanted to check for and we wanted to investigate. So Valeria in the lab, when using this cross-sectional study, she used a self-reported subjective response to alcohol. This is an SRE. It asks about the first 5 times that you drank, how many drinks did it take you to feel something? How many drinks until you stumbled, until you passed out? It asks about the most recent 5 times you drank, the heaviest time that you were drinking, how many drinks did it take you to feel? And what she found is that in bipolar disorder, lower gray matter volume, and again, remember, this was associated with increased risk for problems over time in bipolar disorder, but lower gray matter volume was associated with reporting that they needed less drinks to feel something. The opposite was observed in the typical developing young adult sample. More importantly, again, I'm all about longitudinal data. We always have to be very, very cautious when linking anything as risk or resiliency within subject data. What we found was that when looking 1.5 years later, this increased sensitivity to alcohol in bipolar disorder, not the typical developing group, was associated with an increase in the number of days drinking over time, as well as increased in the quantity of alcohol use over time. And so potentially supporting youth with bipolar disorder may show increased sensitivity to alcohol, and that this increased sensitivity may drive trajectories of alcohol use. So this data was preliminary data for my K01, in which we're starting to test the hypothesis that differences in brain development in bipolar disorder may alter subjective experience of alcohol, neural sensitivity to alcohol, alcohol use patterns, and contribute to risk for alcohol use disorders. So for this study, we bring individuals in, bipolar disorder type 1 or healthy young adults. They're 21 to 26 years of age. Since we are serving alcohol, they have to be 21 to be part of this study. They complete a clinical evaluation, a lot of behavioral phenotyping, and then we put them in the scanner to get some structural resting state data. They then come back on 2 separate days, and then we do counterbalanced placebo-controlled alcohol administration when we either dose them to a 0.08 or we tell them you're receiving alcohol, but we don't give them alcohol. And then during both of those sessions, they complete a lot of behavioral measures as well as complete an fMRI scan as well. That includes both task-based fMRI tasks as well as resting state data. This is just one individual's breath alcohol concentration over the course of their alcohol session. The red just shows you where we're getting subjective response data at, so asking them, how are you feeling? What you can see here is after they start drinking, we take their sort of ascending limb breath alcohol concentration. They go into the scanner during their ascending limb where we do expect more stimulating effects of alcohol. They then peak. We bring them out of the scanner. We also collect subjective response data at the descending limb, 0.06. And then they're allowed to stay into the lab until they hit that 0.04 and they can be escorted home. We time subjective response during the placebo session to the alcohol condition. With one exception, we do not collect this later time point because with the placebo condition, by the time they get out of the scan, the gig is up. They know they haven't had a lot of drink. They definitely feel fine. And so we tell them they hit their exit breath alcohol concentration roughly 30 to 35 minutes when they get out of the scanner that day. This is just one example of one of the subjective response measures that we get. So this is the subjective effects of alcohol scale. And we get this because it has stimulating effects of alcohol, but it also taps into the anxiolytic effects of alcohol, which we thought this could be very important, especially in the context of bipolar disorder. And then we also get sedative effects of alcohol, both positive arousal and negative arousal. So are we seeing differences in the way youth with bipolar disorder respond to alcohol? I am happy to say in my progress reports that yes, we are. So far, we've recruited 25 typical developing controls and 24 young adults with bipolar disorder. We have our 25th bipolar disorder coming in tomorrow. So we're excited to finish out this study over the course of the next few months. And so I'm going to break down this figure for you. So what you're seeing, our black lines are bipolar disorder, young adults, and our orange lines are your typical developing comparison young adults. The solid lines are the alcohol sessions, and the dashed lines are your placebo session. And this is just how they're responding before they got into the scanner and then whenever they're getting out of the scanner. And so what you can see when looking at how lively, talkative, fun, and funny, so stimulating effects of alcohol, while there was an increase in stimulating effects in young adults, healthy young adults in general, you can see that this increase was much more robust in young adults with bipolar disorder. They're reporting much more stimulating effects of alcohol compared to their typical developing peers. Now, one of the things that was most robust, however, was when seeing main effects in bipolar disorder. So for example, we saw that individuals with bipolar disorder felt more anxiolytic effects of alcohol to both their placebo and their alcohol condition, which we found incredibly exciting. And when I show you the drug effects questionnaire on how drunk do you feel, when we saw this, I was completely floored, because this one you can easily see that, yes, placebo and alcohol condition, youth with bipolar disorder report feeling more drunk. And so I find the placebo data incredibly interesting. Keep this in mind because I'm going to circle back to it in a few slides.
Can I ask one question now?
Yes, please.
On the lively talking, fun and funny, or no, relaxed, those are self-reports.
They are.
Did you say that the technicians who were doing these tests also thought they were lively, fun and funny? Or?
No, we've had people also ask us, do you think that this is making individuals more manic? Are they acting more manic during these sessions? No, anecdotally speaking, not at all. And so, right, in all of these, I should also say, this controls for how they felt when they first came in that day. And so we have people fill out these subjective response measures before they start drinking. So that way we can control for sub-threshold changes in anxiolytic effects, sub-threshold changes in sort of lively talkative, since we are studying bipolar disorder, and that's one of the symptoms of mania. And so all of these controls to subtract that out. But in terms of like the eye, no, we're not seeing what we would think would be considered manic-like behavior. We're not seeing something where we would say, oh, you are incredibly talkative compared to someone else when you're dosed to a 0.08. If anything, what you see is, right, people come in and if they're talkative before, they continue to be talkative, but their behavior remains pretty consistent, at least from the RA's perspective.
So it's more of their internal feeling rather than.
Yes, that's a fantastic question. And we did see that changes in the way individuals felt related to their recent alcohol use. So in bipolar disorder, we saw that greater anxiolytic effects during both conditions, the alcohol and the placebo, related to greater recent alcohol use. Conversely, we saw that greater feelings of being drunk, predominantly during the alcohol condition, related to less alcohol use in the typical developing group. So right in the typical developing group, this sort of falls into the low response model. Those that don't feel as drunk, those are the ones that are drinking more. While all of a sudden those in the bipolar disorder group, I would say data is following more in the differentiator model. Specifically, they're feeling more stimulated effects, but specifically the positive effects of anxiolytic reaction to alcohol is associated with greater recent alcohol use. Now, of course, I'm a neuroimager. I'm a neuroscientist by training, so very interested to get to the brain data. I'm not going to go into the task. This is the functional task we're using during the scan. It's a continuous performance task with both emotional and neutral distractors. There's been a lot of data in bipolar disorder with this task, which is why we wanted to use it, including during mania, including during depression, and it reliably elicits activity within the ventral prefrontal systems that we're interested in bipolar disorder. And so the data that I'm going to be showing you is modeling emotional stimuli. And these are negative valenced emotional stimuli that we know will elicit activity within these ventral prefrontal systems. Again, we're taking an ROI approach, ventral prefrontal systems, because we know they're implicated in bipolar disorder. We know they're affected by alcohol, both acutely and chronically. And so do we see differences in the way the brain's responding to alcohol? Yes, we do. This should be considered preliminary. We are in the process of pre-processing our activation data. But what you can see here is that when looking at amygdala response to the emotional stimuli, the black bar here shows you the youth with bipolar disorder. And what you can see is that there is a robust decrease in amygdala activity to these negative valence emotional stimuli in bipolar disorder, but we're not seeing this robust change in the typical developing group. We also see a similar change in terms of looking at anterior cingulate activity in bipolar disorder, in which you see this robust decrease that's not being observed in the typical developing group. What's interesting, again, if we link this back to behavior, decreases in amygdala activity to emotional stimuli was also associated with reporting that they feel more mellow, relaxed during the scan. Now, our most robust findings has really been when doing our functional connectivity analysis. And so my graduate student, this is part of her dissertation that she just defended. She's preparing to submit this for publication now. But what Dylan's finding is that in youth with bipolar disorder, there is a significant decrease in functional connectivity between the amygdala and the ventral medial prefrontal cortex. This is showing you placebo to alcohol condition. That's not observed in the typical developing group. And in bipolar disorder, this significant decrease is associated with feeling more mellow and relaxed during their alcohol sessions. Conversely, Dylan is seeing that in youth with bipolar disorder, there is an increase in functional connectivity between the nucleus accumbens and the ventral medial prefrontal cortex. And this increase in functional connectivity shown here in this black line is associated with them feeling more stimulating effects. So feeling more lively, talkative, fun, and funny. We actually see the opposite pattern in the typical developing group in which they're showing a decrease in functional connectivity between the nucleus accumbens and the ventral medial prefrontal cortex. So in summary, what does this all mean? For us, what we're seeing that it means is that young adults with bipolar disorder, they do show differences in subjective response to alcohol. And this should be considered preliminary, right? We've got roughly 25 per group. We're gonna have 30 per group for this study, but that's still really small for a neuroimaging study. But it provides preliminary support to continue looking in this area as a potential risk factor for subsequent alcohol use problems. We're seeing differences in the way they respond behaviorally as well as the way their brains are responding to alcohol. But we're also seeing differences in the way that they're responding to placebo. And this has fostered a huge interest in thinking about the placebo condition and thinking about alcohol expectancies in the lab. So Dylan, this was actually just accepted for publication. She was very interested in the placebo response as well. And she wanted to look in our typical developing group to see, okay, what happens during this placebo response? What's changing in the brain as someone thinks that they're getting alcohol, but they're not actually getting alcohol? And what she observed was that during the placebo condition compared to before the placebo condition. So remember, during our paradigm, people come in before their beverage sessions, and they complete an MRI scan. And one of those scans was a resting state scan. They also completed resting state scans during their alcohol and their placebo beverage sessions. So she was able to model change in our typical developing group. And she saw that increased nucleus accumbens to the ventral medial prefrontal cortex and the subcolossal cingulate cortex was associated with the placebo response. And specifically, those that showed an increase in the nucleus accumbens to subcolossal cingulate cortex functional connectivity, those are the ones that reported feeling the most drunk during their placebo session. We have a lot of interesting conversations about this data right now. I'm not going to go into it all because we go down some rabbit holes. But if you remember, in bipolar disorder, we're seeing differences in response just to the placebo condition. So we're really excited to explore this more. We're currently doing an analysis analyzing this data to study the placebo response in youth with bipolar disorder. And there's a lot of work that's emerging. There was work in alcohol expectancies, but there's also research recently as interventions, including expectancy challenges, are beginning to be utilized, and they're showing effects in actually changing drinking patterns in youth. And so this is something, right? If you think about where can we take this with bipolar disorder, if there's a way to challenge expectancies, this is something that's really associated with risk. This is something that we can start doing in the clinic with relatively little effort to hopefully have a profound impact. So where do we go from here? Finishing this all up, we're very interested in thinking about how bipolar disorder increases risk for alcohol use disorder. But if you remember neural progression in bipolar disorder, the brain is changing. We see differences in neural trajectories early in the course of illness that's associated with the time in which we see symptoms of alcohol use disorder emerging. Well, we're interested in testing the hypothesis if differences in neural trajectory is associated with emerging and progressive differences in subjective response to alcohol. We're going to be doing a 5-year study following individuals over a 2-year period, longitudinal placebo-controlled alcohol administration to look at development of subjective response, look at the development of alcohol expectancies, the placebo response during young adulthood, and actually do some predictive modeling to see what really is associated with emerging alcohol misuse. And keep in mind, even with this, following people 2 years, We're still going to miss some that's still going to emerge having alcohol use problems, but it gets us a start in following these individuals. Also, it's very important for us to think about as alcohol is being used, alcohol is also affecting the brain. We recently published a study suggesting that prefrontal systems and mood disorders may be more vulnerable to the neurotoxic effects of alcohol, even at low and moderate doses. So, we have people that come into clinic and they say, what is safe to drink? Is 2 drinks safe? Can I do that? Where's the risk really lie? Well, in the context of bipolar disorder, we don't know. And I think that's the key why we need these studies, because it's very true. For some individuals, even low doses of alcohol could be problematic and could result in worse outcomes over time, including being associated with certain neural trajectory patterns. And so this is something that we also want to test. Of course, we know there's other common risk factors, including heritability, early life stress, acute and chronic stress. What are the roles genetic risk and stress is playing? We don't know. We are in the process of finishing up a study that's looking at familial risk for bipolar disorder. It's a 2-by-2 design. So individuals have parents with bipolar disorder or they have parents with alcohol use disorder. So you have individuals, parents with both, individuals with parents with just one or the other, and individuals with no parents with bipolar disorder, mood, or alcohol problems. And what we're doing is we're bringing these people in, and in a simulated bar lab at UT Austin, we're dosing them to a 0.08, or we're giving them placebo, counterbalanced within subject. But again, they're wearing these transdermal ankle monitors. And what's really exciting about these is that they can passively wear these. In future research, they can go home. We can send these to people, and we can see how they're responding to alcohol and have this come in conjunction with EMA methodology to look at how they're drinking, what are they doing, where are you at? This is preliminary. The study was severely delayed because of COVID pause and trying to get approval to have people come into a bar and drink alcohol and take your masks off when everyone was returning from COVID. But right now with the preliminary data, do we see differences based on parental risk? Yes, we do. And we find it incredibly exciting. So just to break these down very quickly, this is showing you alcohol use disorder risk right here on this side of this bar graph. The gray bars mean that they did not have a parent with bipolar disorder. Black bars mean that they did have a parent with bipolar disorder. And what you can see is that individuals with a parent with alcohol use disorder risk, regardless of bipolar parent, yes or no, they showed a lower transdermal alcohol concentration elimination rate. And by this, all that I mean is that their slope of elimination was slower. It took them longer to actually eliminate alcohol during this drinking episode. Conversely, when looking at the bipolar parental risk, again, alcohol risk, no, alcohol risk, yes, bipolar risk is in the black. What you can see is that individuals with a parent with bipolar disorder, they're taking a longer time to reach their peak transdermal alcohol concentration. Now, this is preliminary. We haven't done a lot with this data set yet. We did see that TAC elimination rate related to alcohol use in youth with a parent with alcohol use disorder, suggesting that this could actually be relevant. Still, stay tuned. We'll see what this goes, but I'm very excited about it. We're also interested in early life stress. When looking at familial risk, genetic vulnerability, yeah, there's some there. There's also going to be resiliency that's there. But we also know there's going to be early life stress and differences in early life environment. So it allows us to begin teasing some of that apart and the role that may be playing. We know that early life stress contributes to large risk for both mood disorders as well as substance use disorder. And there was even one study that suggested youth that have comorbidity, over 90, I think it was 95% of them report early life stress, childhood maltreatment. It's incredibly relevant. We can't be ignoring it. Dylan, one of her first papers in the lab was to look at interactions between familial risk for bipolar disorder, childhood trauma, alcohol use. And what she found was that childhood trauma was associated with lower gray matter volume in a lot of these same regions that we see predicts future alcohol use problems, but associated with lower gray matter volume, especially in those with parental risk for bipolar disorder, and especially if they were showing prodromal symptoms, so symptoms of depression and anxiety, which could be suggested that they're at greater genetic risk for bipolar disorder. We also saw that lower activity in those with familial risk for bipolar disorder was associated, not lower activity, lower structure, lower gray matter volume, was associated with greater drinking, but only in those in familial risk. right? And so again, it points to early life stress by familial risk, genetic interactions that may drive or contribute to some of these outcomes. And since some of these regions are the same regions that we see are related to variation in subjective response to alcohol, we're interested in what is early life stress? How does this relate? Is there any support for early life stress may relate to subjective response? Yes, not in the context of alcohol. There has been studies looking at amphetamine, seeing that early life stress related to increased pleasurable effects of amphetamine exposure. Raquel Costed in the lab right now is working on this analysis. This is only looking at healthy individuals, but healthy individuals that have parents with mood substance use disorder compared to those that do not. And she's also seeing that greater childhood trauma scores on the childhood trauma questionnaire is associated with them reporting that they feel more drunk compared to those that do not have childhood trauma, or I should say those that do not have a parent with substance use disorder. Again, suggesting early life stress by familial risk interactions that may drive some of these behavioral changes that we think could be a risk factor. I'm going to really speed through this now. We're interested in stress, both chronic, acute. There's reason stress is implicated right in addiction a lot. It's also implicated in bipolar disorder. Work suggests that stress increases sensitivity to alcohol. This is really preclinical work. Joyce Bashir at Yale has been a huge force in this field in thinking about this. And while very few studies have looked in humans, we do know that cortisol response is related to variation in subjective response to alcohol. We've done some preliminary work in this, thinking about how perceived stress relates to how drunk people are getting, even when controlling for sex, the number of times people binge. And we are seeing stress does seem to map on to variation and self-report how they're feeling. And when we also look during our alcohol sessions, we are getting a weak signal there that mirrors this. And so this is something that we hope to be able to investigate with this future longitudinal grant. So in summary, we know it's complicated. It is noisy. There's a lot going on. But when thinking about risk factors, we can't think about one risk factor independently. These factors are interrelated. They are interacting with one another to contribute to these outcomes. And so we do need larger data sets. And luckily, knock on wood, it does seem like NIAAA is supportive in that effort at this point. We are seeing that familial risk interacts with early life stress, specifically familial risk, I should say, not necessarily genetics. We collect samples, but we have not done any genetic analyses of these samples. We're seeing that these interactions may contribute to variability in neurodevelopment of the brain. This variability in neurodevelopment of the brain may contribute to alcohol, marijuana use, and associated outcomes. And especially through differences in stress and alcohol response. And for us, we don't think, right, stress and reward systems, they're not independent. We know that they interact. And so we think it's incredibly important for us to be thinking about these systems in concert and how it's shaping risk and changing over time as alcohol misuse emerges. And hopefully the goal is to be able to target these factors for interventions. But with this, I will stop. I really want to thank my lab. They've been phenomenal. None of this work would have been possible without them. I especially need to thank Dylan, Raquel, Vanessa, and Valeria, who did the bulk of the heavy lifting for these projects. Of course, my collaborators at UT Austin, and also my funding sources. And of course, the participants who gave us a ton of feedback as we were getting these studies up and running and have been incredibly supportive. And with that, I'll stop and take any questions. Thank you.
When people ask questions, repeat the gist of the question so that people on the Zoom can.
Yeah, I'm actually going to move a speaker on this table, so if you guys don't mind, I know it's a little awkward. Just kind of like a town hall. Yes, if you don't mind. Let's see, I know, I'm so sorry.
I feel like I'm on like or something. Sorry if this is a silly question. I was wondering, just as you were talking about, have you thought about diet or how body weight might be affecting them? Because I'm thinking about how depression and mania may affect their eating patterns, and adolescence is such a big developmental period, and how that may be affecting both bipolar disorder, but also how they're subjectively affecting the alcohol.
Oh, Katie, you make it difficult. Yes, we have, but not enough. I will say that, we collect BMI. We control for that in studies. When we're dosing people to a 0.08, we are looking at weight and height to help dose individuals. But based on metabolism and body type, beyond weight and height, muscle versus fat content, that can change alcohol metabolism. And we are not actually assessing that. And so that's a limitation that we need to do better in future studies. But in terms of thinking about symptoms, all individuals here are euthymic, although you do see greater subthreshold depression and anxiety symptoms. We control for anxiety symptoms in all of our models. Because of that, depression and anxiety were highly correlated within the sample. So we just went with anxiety over depression. But we do see variability in when people last ate. We tell people eat 4 hours before you're going to be starting to drink. So they know that. They come in and we give them a weight adjusted pretzel. So for every pound, you're going to eat a pound, or you're going to eat 1 calorie of pretzel. So everyone has the same amount on the stomach. But people do not do that. Everyone will wait 4 hours, but there are times in which someone comes in and you ask them, when did you last eat? Oh, last night sometime. It was around, it was like, oh, you didn't eat anything this morning, right? And there's nothing we can do to control that. And so that does add another degree of noise to the data set, in which now we're trying to be very, very cautious about trying to get when you last ate, how much you ate, so we can try to control for some of that noise, because that can have a huge impact on the way individuals are experiencing alcohol. As everyone knows, I remember in college, you knew if I'm going to go have some drink, I need to get something to eat first. I can't do that. Maybe, my friend can. There's no way I can manage that. And so I think that's one of those things where it can definitely have a profound impact. And that could be something that drives variability in response to alcohol. So that's something I'm really interested in, is thinking about, sort of stable dynamics of alcohol response compared to unstable dynamics. And if you sit there and you think about learning and memory, and you think about alcohol expectancies, and you think about, you experience something and that updates your memory, which, okay, when I drink this much, this is how I'm going to respond. And so this is going to then affect my behavior later on. But then if you're seeing stress relates to differences in the way you're responding to alcohol, if you're seeing that food intake is relating to differences, if you're not in tune with that, how are you updating, this learning, this memory, and how is this driving your behavioral adaptation, not only to the way you're viewing alcohol, but the way that you're actually engaging with alcohol? And I think this is something really important that we have not done a lot of work in, because a lot of the literature, they do look at alcohol responses being stable. And if you look at Andrea King's work that emerged roughly a year ago, she challenged the complete idea of tolerance as a risk factor of alcohol use disorder when she followed individuals over a 10-year period and saw these individuals with alcohol use disorder, including increased severity. We're seeing that they're reporting increased sensitivity to alcohol, not decreased sensitivity, but increased sensitivity. And it gets more to stress sensitization, thinking about how alcohol is affecting these systems. And so it's really attractive data that points to, we don't know exactly what's happening and what the risk factors are, but thinking about some of these really important factors that could be contributing to this is incredibly important. And thinking about, where do we go from here? How do we intervene? How do we, educate people about what these risk factors are? And right now we can't, because we don't know, but hopefully we can start to uncover some of them. Great question.
Good questions?
All right, these are thoughts that I'm putting together on the fly, so.
I like that.
You probably thought about it more than me. So I'm thinking of this slower clearance of alcohol and this lesser sensation in response to the alcohol. And I'm wondering if acute tolerance is playing some role there, and you started to touch on it in your previous answer. So just tell me more about that.
Yeah, and it's interesting. So, in the youth with parents with alcohol use disorder, we did see that their slower clearance was related to alcohol use. And if I'm remembering the directionality of the relationship, it was that slower clearance specifically related to not having drank as much recently. And so, it's a little bit opposite of what we would hypothesize. I didn't show it because I don't understand it and because it's still preliminary. But that is one of the things where, and I think a lot of the things that we're seeing in our healthy development, they do sort of foster this idea a little bit more of tolerance, where previous exposure is associated, and it makes sense, if someone's been drinking they may have a faster alcohol metabolism, they may be metabolizing it faster, so they're able to drink more compared to someone that hasn't been drinking as much. And so you could follow that under the tolerance umbrella. However, in the bipolar group, we're not seeing any evidence of that, right? And so in them, we're actually seeing that, yes, it's taking longer for them to reach their peak transdermal alcohol concentration. But for them, right, that doesn't relate to their recent alcohol use. There's something else potentially going on there. So I think as we think about risk factors, I think we also need to be thinking about factors such as tolerance. Factors, well, what is recent alcohol use doing in this group that it might not be doing in other groups? And thinking about how adolescents, alcohol and substance use, how that might differ in terms of the negative consequences, or let's just take negative, just in terms of the associations with brain development, with brain health. And it may differ by some groups. Because we were even talking earlier, right, that there's some individuals, and I can't remember who we were talking about, that can, I think it might have been you, Katie, but right, they can, drink a lot and then they're walking around and they're completely fine, right? And then there's other individuals that can't. And I think some of these individual variabilities just in the developing group and, other groups with vulnerability, I think it's incredibly an intriguing sort of idea of thinking about interactions between exposure and risk factors and what's driving changes in the development of tolerance of whatever put in other context A or behavioral outcome A may be actually driving these, which is something that we need to look more into.
I had a question in terms of medication. I may have missed it in terms of the individuals in the study who were diagnosed with bipolar. Are they receiving medication for their, yeah?
They are, yes. And so we do not exclude for medication.
Yeah.
We have a clinical team, including a pharmacist that's independent from the study, that reviews all the medications that an individual is taking before they can do their alcohol sessions. And that's part of our IRB, our safety measure, because we knew we're not going to be able to recruit anyone if we exclude medication.
Right.
Individuals with bipolar disorder, they drink regardless of their medication, and so it's still an important question. But we have explored within this data set effects of medication, which we think is incredibly important. And while right now it is incredibly underpowered, we are seeing that some medications may be associated with variation in subjective response to alcohol in youth with bipolar disorder. So for example, we saw that sedatives being on board, sedatives, antihistamines, was actually associated with youth with bipolar disorder feeling less woozy, drowsy. And it was actually the opposite of what we would have thought. Yeah. Right. And then we also saw something with anticonvulsants, in which anticonvulsants, those also were associated with variability in subjective response to alcohol, although I'm not remembering which specific subscale it was associated with. And so I think it's really important, right, as we're sitting there, we're thinking clinically about risk factors. And if we have someone that's coming in, we also need to know more how medications are affecting some of these behaviors, because that could be really important in terms of thinking about prescribing certain medications to some individuals and trying to map with their particular phenotype or risk or resiliency profile to think about what might have the best outcome or decrease risk for developing problems over time.
Okay, and then certain other drugs that maybe not be prescribed but taken, like caffeine, cannabis, nicotine. Do you have any data or ideas about these?
Yeah, it's a great one. So I think the comorbid cannabis and alcohol is really interesting. We do see more individuals, right? We know cannabis is widely used by healthy youth as well as by youth with bipolar disorder. We're not seeing a relationship with cannabis in this study. We don't have a lot of people smoking. We do have a few more that actually smoke, but we're not seeing a lot of people that we're recruiting right now that are actually smoking and using tobacco products, which is a little surprising. We did expect to see more. We don't see the other drugs on board. But that's also an exclusion criteria. Individuals, they can have a cannabis use disorder that's mild or moderate. They can smoke, but if you're, if you have cocaine use disorder, or if you have other problems with other drugs, you're excluded. Try to minimize the noise, in which we embrace the noise, but at the same time try to minimize it, at least in a few places.
Okay, thank you.
Yeah, thank you.
Probably need to wrap up. Thanks so much. It was a wonderful talk. It looked like there was at least one comment. Maybe Christina Mead was just saying what a great talk it was.
Yes, she was saying very impressive work. Fantastic.
So good. So maybe you'll hang around a few minutes if people want to chat on the way out. Very good.
Definitely.
Okay.
Yeah, thank you guys.
Thank you.
January 12th, we're going to start the spring 2023 CVAC seminar series here. Again, it's going to be on Thursdays, 2 to 3. Christina Mead's going to speak.
Go back to